Hello everyone, my name is Jeong. It's a great honor to have an opportunity to present my research in this time. Today, I will present you on the reduction in the biomass of nitrogen fixer and the biological pump in the Northwestern Pacific Ocean. The role of the ocean in the global carbon cycle is important for the potential uptake of atmospheric carbon dioxide. In particular, phytoplankton in the surface ocean converts inorganic carbon to organic matter during photosynthesis. A part of organic matter is exported from the surface ocean and reaches the deep ocean. This process is referred to as the biological pump and is a main mechanism that sequesters atmospheric carbon dioxide to the deep ocean. Global warming studies suggest that the biological pump will be reduced in the future ocean because global warming will enhance ocean stratification, so reducing nutrient supply to the surface ocean. However, nitrogen fixation will relieve this nutrient limitation in the future ocean because nitrogen fixation transforms inert nitrogen gas to bioavailable new nitrogen in warm, oligotrophic, and stratified waters. Then, nitrogen fixation will be one of the key processes to determine the biological pump in the future ocean. To verify this hypothesis, it is necessary to evaluate the response of nitrogen fixation to global warming and its impact on the biological pump. The objective of this study is to evaluate the response of nitrogen fixation to global warming and the impact of changes in nitrogen fixation on the biological pump in the Northwestern Pacific Ocean. For this, we investigated the relation between sinking particle flux and nitrogen fixer biomass and compared the results with physical and biogeochemical parameters. Sediment trap experiment was conducted from 2009 to 2014 at station FM2. Sediment trap was deployed at depths of 1,000 meters and trap samples were collected at intervals of 18 days. The samples used to determine the fluxes and nitrogen isotope value of particles. Biogeochemical data were obtained from our springtime field survey, and physical data were obtained at the website. Our sediment trap data are presented in this figure. In 2009 and 2010, particle flux showed a distinct peak during warm season. However, this peak has almost disappeared since 2011, and particle flux had a decreasing trend during the observation period. Vertical bar indicate particle flux and green line indicate chlorophyll A concentration. Particle flux changes seasonally. This seasonal pattern was broadly consistent with chlorophyll A as the seasonality in chlorophyll A concentration, suggesting a coupling between primary productivity and particle flux. Furthermore, nitrogen isotope value of particles were generally lower than the value of deep water nitrate in the subtropical Pacific Ocean, indicating that nitrogen fixation contributed significantly to primary productivity in all seasons. Consequently, particle flux may be highly associated with the nitrogen fixation in all seasons. Particle flux showed an annual decreasing trend. To eliminate the seasonal effect, annual evolution of the particle flux was separately made on 4 seasons. Particle flux significantly decreased during spring and summer. Since our field data were obtained only in spring, we focused on the springtime flux decrease. What led to the annual decrease in the springtime particle flux? We observed the annual decrease in both nitrogen fixer abundance and depth integrated chlorophyll A concentration. Considering that nitrogen fixation contributed significantly to primary productivity in the study area, decrease of nitrogen fixation resulted in the decrease of primary productivity and hence particle flux. Then, what brought about the decrease of nitrogen fixer abundance in spring? The growth of nitrogen fixer depends on several factors, including water temperature, nitrate, ion, and phosphate. In the study area, generally high seawater temperature and nitrate deficiency conditions were favorable for the nitrogen fixation during the entire year. So these factors could not constrain the growth of nitrogen fixer. We investigated the temporal changes in the atmospheric ion input using aerosol concentration. The noticeable trend in the springtime aerosol concentrations were seen during study period. In addition, several studies reported that ion did not act as a limiting factor for the nitrogen fixation in the Western Pacific Ocean. Therefore, ion was not main driver for the changes in the nitrogen fixer abundance. During field survey, we observed annual decrease in the phosphate inventory. It is well known that phosphate is a major factor for the controlling the growth of nitrogen fixer. Therefore, decrease in the phosphate inventory may result in the decrease in the nitrogen fixer abundance. We further investigated the reason for the reduction of phosphate concentration in spring. As SST increased linearly, the springtime mixed layer depths decreased significantly. This decrease in the mixed layer depths may have decreased phosphate supply from deep ocean, thereby reducing the activities of nitrogen fixer. In summary, we reported the 5-year reduction in biological pump in the Northwestern Pacific Ocean, which was noticeable during spring and summer. Our springtime field survey shows that decrease in nitrogen fixer abundance, thereby reducing new nitrogen supply to surface ocean, mainly caused the weakened biological pump. Increasing SST and decreasing mixed layer depth might trigger the decreased phosphate supply to the surface ocean. Our result differs from that observed at Northeastern Pacific Ocean. KIOST shows that nitrogen fixation had been enhanced during a period of prolonged stratification at Station Aloha. It seems likely that the response of nitrogen fixation to enhanced stratification may change significantly over the global ocean. Hence, it is vital that we conduct more studies to evaluate the response of nitrogen fixation and the biological pump to environmental changes in as many ocean settings as possible. Thank you for your attention.
Thank you, Dr. Jeong.